Inside every cell of your body lies DNA, the master instruction manual that controls how cells grow, repair themselves, and communicate. When this code remains intact, your body functions normally. But when DNA becomes damaged and those errors go unrepaired, cells can begin to behave abnormally. This is where cancer often begins. At the ends of every DNA strand are structures called telomeres. Think of them like the plastic tips at the ends of shoelaces. Their job is to protect the genetic code from unraveling. Each time a cell divides, telomeres become slightly shorter. Eventually, when they become critically short, cells can no longer divide safely. This contributes to aging and increases the risk of genetic instability, one of the earliest warning signs in cancer development. But what causes DNA damage in the first place? DNA is constantly exposed to stress from both inside and outside the body. Environmental toxins, pollution, smoking, excessive alcohol use, poor diet, obesity, chronic inflammation, and prolonged stress can all cause breaks and errors in the DNA. Normally, the body repairs this damage, but when repair systems fail, mutated cells may begin dividing uncontrollably. Another critical player in this process is the mitochondria, the power plants of the cell. Mitochondria generate energy in the form of ATP and help regulate when damaged cells should self-destruct through a process called apoptosis. In cancer cells, mitochondrial function becomes disrupted. Instead of producing energy efficiently using oxygen, cancer cells shift to a much less efficient method called fermentation. This metabolic shift, known as the Warburg effect, allows cancer cells to rapidly produce energy from glucose. However, this process generates large amounts of lactic acid. The resulting acidic environment damages surrounding tissues, suppresses immune responses, and helps tumors grow and spread. Healthy cells rely on oxygen and mitochondria to produce clean energy. Cancer cells avoid this pathway and instead depend heavily on sugar metabolism. Another critical defense against cancer is a gene called p53. Often referred to as the guardian of the genome, p53 monitors DNA damage and determines whether a cell should repair itself or undergo programmed death. When p53 becomes damaged or disabled, this protective system fails. Cells with dangerous mutations can survive and continue dividing, one of the key steps in cancer progression. Scientists are now discovering new ways to detect and monitor cancer earlier than ever before. One promising technology is the liquid biopsy, which measures circulating tumor DNA, or ctDNA, in the bloodstream. These fragments of cancer DNA provide real-time information about tumor activity, treatment response, and emerging resistance mutations, all through a simple blood test. Understanding how DNA damage, telomeres, and mitochondria interact is changing the future of cancer research. By targeting cancer metabolism, restoring cellular repair systems, and identifying key genetic mutations, researchers are developing more precise and personalized strategies to fight the disease. The battle against cancer is happening at the smallest level, deep inside our cells, where DNA, energy, and cellular survival pathways determine the outcome.